Then there comes the passive immunity. In passive immunity, the quick this is a quick but a short-term protection against an infection can be provided by the administrating the preformed antibodies, and these antibodies may be obtained from animals and obtained from human beings or synthesized in the lab. And these monoclonal antibodies produced in the laboratory are done through the hybridoma technology, which are widely used for the academic purposes, diagnostic purposes, and the therapeutic purposes. Here, how this production of the monoclonal antibodies take place, and these monoclonal antibodies are the clones of each other. They are the only one type of antibodies. They are generated in the huge amount, and they are produced in the laboratory by a hybridoma technology. A hybridoma cell is a hybrid cell which are prepared by fusion of B lymphocytes and the myeloma cells. These are the cancerous cells. B cells are fusing with the cancerous cells. Then these B lymphocytes are antibody forming cells, and a given B cell lymphocyte is dedicated to form the one particular antibodies. And whereas the myeloma cells are the immortal cancer cells, and fusion of the B lymphocyte with the myeloma cell produce the hybridoma cells. And these hybridoma cells possesses properties of the myeloma cells as well as those of the B cell. So the hybridoma cell is an immortal like a myeloma cells, but it is synthesizing one particular antibodies that is given by using a B cells. So this hybrid form is very much important in hybridoma technology for producing the only one specific of antibodies, and when this hybridoma cell is put in the culture medium, it goes on dividing forever, because the cancer cells having the property of dividing continuously. Whereas along with the dividing, they are also secreting out the antibodies that is required. Since the only one lymphocyte has been fused with the myeloma cells, so the hybridoma cells are secretes the monoclonal antibodies. And how this take place, some of the procedure is given here. The mouse is firstly incubated with the antigens that is needed means the through which we are getting the antibodies that is required, and the B cells are taken from its spleen. In the B cells, the activation takes place against that of the antigens, and B cells are activated, and these B cells are isolated, and these B cells and cells are fused with the myeloma cells in the presence of the polyethylene glycol, which is the fusing agent. And when these B cells and the myeloma cells are put together in presence of the PEG, some B cells may fuse with the B cells, and some the myeloma cells may fuse with the myeloma cells. There are the various possibilities that take place inside the fusion process. And some B cells may fuse with the myeloma cells, and these are combinations. Some B cells and the myeloma cells may be remain unfused also. And when the fusion of the B cells with the myeloma cell produce is the hybridoma cells, where the hybridoma cells have to be selected from the other cells. Now, how do we select these hybridoma cells from the other cells? For this is done by the culturing the cells in the hypoxanthine aminotransferrin thymidine, that is HAT medium. This HAT medium provide us to differentiate between the hybridoma cells and from the other cells. Here the systemic representation of this production of the monoclonal antibodies. First of all, there is the injection of the antigens inside the mouse model, and this mouse, then the spleen are taken out, and they are then fused with the myeloma cells, and these fuse leads to the different population of the cells of the fused and the unfused and the hybrid cells. These are then selected using the HAT medium, and this only select the hybridoma cells. And these myeloma cells lack the HGPRT, and therefore they cannot perform the salvage pathway and this in the presence of the head video, and they are dependent on the de novo synthesis for their purine requirement. But de novo synthesis is blocked by the aminopterin. So in head medium, the infused B lymphocyte die soon when their lifespan is over, and this is how the B cells are separated out. The infused B cells die out soon when their lifespan is over, and the infused myeloma cells cannot survive because they are deprived of the purines. And, also the myeloid cells are removed out. The only hybridoma cells survive as they have acquired HGPRT from the B lymphocyte, whereas the myeloma cells are lacking the HGPRT. Here, there is a de novo synthesis of the purine nucleotide, and the TMP is blocked by the aminopterin, whereas the myeloma cells cannot salvage hypogentin as they lack the HGPRT. So, the production of the antibodies, monoclonal antibodies take place through these processes.